Okay, for histology notes, for muscle tissue lecture, Dr. De La Peña. Topic outline: skeletal muscle, cardiac muscle, smooth muscle, and the regeneration of muscle tissue. Muscular system, it consists of all muscles of the body. It makes up approximately 42% of total body weight and composed of long, slender cells known as fibers. Fibers are different in lengths and vary in color from white to deep red. Your muscle tissue is composed of cells that are highly specialized to shorten in length by contractility. They are also known as myocytes and are elongated in one direction. It is in mesodermal in origin and is specialized cells which shorten in length contraction, and this is how they bring about movement. It contains contractile proteins, mainly actin and myosin. Each muscle consists of a group of fibers held together by connective tissue and enclosed in a fibrous sheath fascia. Your sarcolemma is the cell membrane or your plasmalemma. Sarcoplasm is your cytoplasm of muscle cells. Reticulum sarcoplasm or your sarcoplasmic reticulum is equivalent to smooth endoplasmic reticulum. It surrounds the myofilaments and forms a meshwork around each myofibril. It forms a pair of dilated terminal cisternae and regulates muscle contraction by sequestering calcium ions, which leads to relaxation, or releasing calcium ions, which leads to contraction. Your sarcosomes is your mitochondria. Now, parts of a muscle. Your body is the main portion. Origin is the fixed attachment. Insertion is point of attachment to the part of the body moved by this muscle. Tendon is the means of attachment, connecting the muscle to the bone. It varies in length from less than 1 inch to more than a foot. Now, your skeletal muscle is a striated muscle. It is voluntary and mainly in limbs and in relation to the body wall. It is elongated, cylindrical, and is multinucleated, which the nucleus are located at the periphery or at the sides. It varies in length from few millimeters to few centimeters. It is alternating and from dark to light bands in cross striations. It mostly originates from somatic mesoderm or from embryonic origin. The basic unit is the long cylindrical fiber, and fibers are arranged parallel to each other. It is formed by fusion of multiple myoblasts during embryonic life. It has dark eosinophilic cytoplasm due to myofibrils, and sarcolemma forms deep tubular invaginations or titobules. Your titobules extends deep into the sarcoplasm. Myofibril are contractile elements. Your sarcomere is the region between two adjacent Z-discs. It is a precisely ordered collection of myofilaments thick and thin. It is the fundamental contractile unit of contraction. Your myofilaments contains your thick, which is your myosin, and thin actin filaments. It lies parallel to the long axis of the myofibril and is responsible for the sarcomere bonding pattern. For general architecture, skeletal muscle fibers are supported by connective tissue framework. First, your epimysium is the entire muscle. It forms aponeurosis that connect skeletal muscle to skeletal muscle. Perimysium is the bundles of muscle fiber. Your fascicles are small bundles of muscle cells and is surrounded by your perimysium. Endomysium is individual muscle fiber. It is composed of reticular fibers and external lamina. Skeletal muscle cell types. First is your red or type 1. It is slow in contraction and does not contract slowly and does not fatigue easily. The white type contracts rapidly but easily gets fatigued. White is your type 2B. Your intermediate is type A, which has the characteristics that resemble both type 1 and type 2. Now, your satellite cells are regenerative myoblasts-like cells. These are residual cells from embryonic development that lie within the external lamina or basal lamina of the skeletal muscle cells and adhere to these cells via M-cadherin molecules. To form new skeletal muscles, first, damage to muscle tissue or mechanical stress activates the satellite cells. Activated satellite cells under the influence of myogenic regulatory factors like MyoD becomes myoblast. Now your myoblasts fuse with each other to form myotubes. Then your myotubes differentiate into mature skeletal muscle. Myostatin is a member of tumor growth factor beta family, beta superfamily. It ensures that muscle does not become overly long or broad, thus restricting the size of the individual skeletal muscle. Now let's go to the striations of skeletal muscle. For your A band, it has a light zone, H zone, and the center of H band is your M line. While your I band is divided into two equal halves by a Z disc. Your A band is anisotropic with polarized light, while your I bands are isotropic. Your A bands are darkly stained, while your I bands are lightly stained. Your A band contains both thin and thick filaments that overlap and interdigitate. 6 thin filaments around each thick filament. Your H band again is the light regions transecting A band. It consists only of thick filaments. While your M bands, it is a narrow, dark region at the center of H bands formed by several cross connections or M bridges at the centers of adjacent thick filaments. Your Z discs or your Z lines are dense regions dissecting each I band. It contains alpha-actinin and GAP-Z, the two proteins that bind the thin filaments and anchor them to Z disc with the assistance of nebulin. Your T tubules, or your also known as transverse tubules. These are tubular invaginations of the muscle cell membrane that penetrate deep into the sarcoplasm and surrounds myofibrils in such a manner that at a junction of each A and I band, these tubules become associated with dilated terminal cisternae of the sarcoplasmic reticulum and forming triads. Now, your terminal cisternae encircles myofibrils at the junction of each A and I band. The triad is composed of T tubules in the middle and the terminal cisternae in both sides, located at the AI junction and help provide uniform contraction throughout the muscle cell. Desmin with vimentin during embryonic development, aided by plectin, anchors Z discs to one another. It is peripherally located. A peripherally located Z discs are anchored to regions of sarcolemma, known as costomeres, dystrophin-associated glycoprotein. Your costomeres are a specialized area of sarcolemma located at regions that corresponds to Z lines of myofibrils that are beneath the striated muscle cell membrane. It is defined by the presence of peripheral and transmembrane protein complexes whose principal moieties are localized on the sarcoplasmic aspect of the striated muscle cell membrane. Dystrophin-associated glycoproteins. You have 5. First, dystrophin. It is a pencil-shaped molecule that is between the ECM and the cytoskeleton attached to the periphery of the myofibril. It plays the greatest role in strengthening and maintaining the integrity of the sarcolemma during the process of contraction. It acts as a link between the cytoskeleton and the ECM. Cycloglycan complex forms attachment to and then reinforce the dystroglycan complex. Your dystroglycan complex is a complex of two transmembrane proteins whose extracellular domains bind to laminin and laminin of the external lamina. Intracellularly, it binds to dystrophins as well as syntrophins. Your syntrophins are relatively small proteins, approximately 60 kilodaltons, and forms bonds not only with dystrophin but also with desmins and distrobivin. Your distrobivin has binding sites with syntrophins and with desmin. These function affix the desmin intermediate filaments to ECM laminin, providing them with the ability to maintain the precise orientation and arrangement of Z lines and of adjacent myofibrils. This attachment occurs in the costamere region of the sarcolemma. Myofilaments molecular organization. Your thin filaments, first you have your F-actin, which is a polymer of G-actin monomers arranged in a double helix, where each monomer possesses an active site that can interact with myosin. It exhibits polarity, having positive having a plus that is tethered to cap Z of the Z discs, and the minus end, which is with the growing end of the F-actin, and is located at the H band and is capped by tropiumodulin. Next, for your thin myosin is your tropomyosin. The tropomyosin they bind to tail, forming filaments that are located in the grooves of the F-actin helix. Next is your troponin T. T for tropomyosin forms the tail of the molecule. It functions in binding the troponin complex to tropomyosin. Troponin C, C for calcium, possesses 4 binding sites. For calcium, it may be related to calmodulin. Lastly, for troponin, you have your troponin I, or for I for inhibitory. It binds actin to actin and inhibits the interaction of myosin and actin. Last, for the thin filaments, you have nebulin. It's a long inelastic protein. Two nebulin protein molecules, wrap around each thin filament and assist in anchoring it to the Z-disc. Next, myofilament is your thick myofilament. It is composed of approximately 250 myosin 2, arranged in an antiparallel fashion. Myosin 2 is composed of 2 identical heavy chains and 2 light pair of light chains. A heavy chain consists of long rod-like tail and globular head. The tail wind around each other in a alpha helical configuration. It functions in the self-assembly of myosin molecules into bipolar thick filaments, whereas the actin binding sites of the heads function in contraction. Trypsin cleaves myosin into light meromyosin, part of the tail portion, and heavy meromyosin, and 2 heads on the remainder of the tail. Papain cleaves the heavy meromyosin, releasing the short tail, which is your S2 fragment, and 2 globular heads, or your S1 fragments. The S1 fragment have ATPase activity but require interaction with actin to release the non-covalently bound ADP and inorganic phosphate. Myomycin and creatinine kinase are located on M-line. Your myomycin affixes adjacent thick filament to one another, and creatinine kinase removes a phosphate from creatinine phosphate to phosphorylate ADP, converting it to ATP. Next is C protein. Your C protein occupies the C zone and binds the thick filaments between the M-line and the end of the thin filament in the vicinity of AI junction. Last is titine, which is a large linear protein that displays axial periodicity. It forms elastic lattice that parallels the thick and thin filaments. 2 titine proteins anchor each thick filaments to each other. The amino terminal spans the entire thickness of the Z-discs and binds alpha-actinin and Z proteins. The carboxyl terminal spans the entire M-linea and binds the protein myosin. Skeletal muscle contraction involves binding, hydrolysis, and release of ATP. For Huxley filament model, during contraction, thin filaments slide past thick filament and penetrate deeper into the A band. However, the I bands and H bands shorten as Z discs are drawn closer. Second, neither the thick nor thin filaments become shorter. Instead, they increase their overlap. Next, skeletal muscle contraction is the imitation regulation of contraction. First, there is depolarization accompanied by the release of calcium, which triggers the bind of actin and myosin, leading to muscle contraction. Sarcolemma is depolarized at the myoneural junction, and the wave of depolarization is conveyed to the myofibrils by T-tubules. Calcium is released into the cytosol of the AA junction via calcium release channels of the SR terminal cisternae. As long as calcium level is sufficiently high, the contraction cycle will continue. Second is the activation of actin via calcium. In the resting state, the myosin binding sites on the thin or actin filaments are partially covered by tropomyosin. Also, TNI is bound to actin. The bind to actin hinders myosin-actin interaction. Now, when calcium is also bound by TNC, resulting in conformational changes that breaks the TNI-actin bond, tropomyosin shifts its position slightly and uncovers the myosin binding site, which is your active state. Now, after the conformational change, there will be contraction cycle. First, the S1 fragment of myosin 2 releases the inorganic phosphate, but the ADP remains attached to the S1 fragment. The release of inorganic phosphate causes conformational change, leading to the strong bond between actin filament and the S1 fragment. The ADP molecule is released from S1 fragment of myosin 2, which results in an alteration of the myosin 2 molecule, and the thin filament is pulled toward the middle of the sarcomere. This is known as the power stroke. An ATP molecule becomes attached to the S1 fragment, resulting to its release. The ATP is then hydrolyzed and the weak bond is formed between the actin filament and the S1 fragment of myosin 2. For a complete contraction in which a sarcomere is shortened by approximately 1 micrometer, the cycle must be repeated approximately 200 times. Now, next is relaxation. Relaxation depends on calcium pump and the SR, which pumps calcium from the cytosol to the inner surface of the SR membrane to be bound by calsequestrin. When calcium concentration in the cytosol is reduced enough that TNC loses its bound to calcium, relaxation occurs as anthropomyosin returns to its resting position. Next is the motor unit. It consists of a neuron and every muscle cell it innervates. The all or none law, it states that either a muscle cell contracts or does not contract at all. A muscle may contract with varying degrees of strength because only some of the muscle cells contract, but an individual muscle cell must obey this law. Now, concentric contraction. As muscle contracts, the sarcomere shortens and the entire muscle becomes shorter. For your isometric contraction, sarcomeres do not shorten and the entire muscle remains at the same length. Now, for your nerve supply. Nerve supply is from motor fibers, which is voluntary, aka the myoneural junctions. Synapse between a branch of motor nerve axons and skeletal muscle cells. The structural components are: the axon terminal contains mitochondria, synaptic vesicle, which contains the neurotransmitter acetylcholine, SER element, Schwann cells, but lacks myelin on its non-synaptic surface. Perisynaptic membrane is on the synaptic surface of the axon terminal. Next is the axon cleft, and it is a narrow space between the presynaptic membrane and the postsynaptic membrane, also known as motor end plate. It contains the amorphous external lamina and a basal lamina-like membrane. Last is the muscle cell near the myoneural junction. The junctional folds is the sarcolemma and vaginations of the postsynaptic membrane. Next for the nerve is the sensory fibers that is responsible for the sense of position and proprioception. The two types are: you have muscle spindle, which is elongated falciform sensory organ within the skeletal muscle that functions primarily as a touch receptor. Normal skeletal muscle cells, like the extrafusal fiber, surrounds it. The capsule of the muscle spindle anchors it to the perimysium and endomysium of the extrafusal fiber. Its functions are stretching of the muscle. It responds to both the rate or phase of phasic response and duration or tonic response. The polarization of gamma efferent neurons stimulates intrafusal nerve endings. Muscle overstimulation results from stretching at too great frequency or for too long at a period of time. Overstimulation causes firing at the alpha efferent neurons and initiating muscle contraction, thus counteracting the stretching. Second is the Golgi tendon organs. It is composed of encapsulated collagen fibers that are surrounded by terminal branches of the type 1B sensory nerves, where they counteract the effects of muscle spindles. It is located in the tendons and is stimulated when the muscle contracts too strenuously, increasing tension on the tendon. Now, clinical considerations. Rhabdomyosarcoma is one of the most aggressive neoplasm involving skeletal muscle, usually restricted to the deep muscles of the arms and legs. It is subdivided into clusters by fibrous connective tissue septa, and when cells in the center of each cluster die, the empty space gives the impression of alveoli of the lung. Possibly, this condition arises as a function of chromosomal translocation. Alveolar rhabdomyosarcoma occurs in young adults. For treatment, surgery followed by chemotherapy and sometimes radiation. Prognosis is much better in children. Next is your anabolic steroid. It is used among athletes and high school aged students to increase their muscle mass. Forms: you have your tablets and injections, but tablets may cause jaundice and liver damage as well as anemia. It is detectable in the body fluids for 6 months after the last dose. Next is your dystrophies, which is a varied constellation of familial conditions that may become evident in preteens or early teens as weakness in muscle group that progress with time to a wasting condition. Biopsies of the muscle groups display the presence of fibrous and fatty tissue in place of skeletal muscle. Duchenne muscular dystrophy or DMD is caused by a sex-linked recessive genetic defect that results in the inability to synthesize dystrophin. This common serious degenerative disorder occurs in young men and results in death, usually before 20 years of age. It is characterized by the replacement of degenerating skeletal muscle cells by fatty and fibrous connective tissue and may affect the cardiac muscle. Your scapulohumeral muscular dystrophy or your FSHMD or your Landau-Digerin syndrome is one of the most common form of muscular dystrophy. It is not sex-linked but is an autosomal dominant genetic defect that results in wasting of the skeletal muscle of the face, those around the scapula and those of the upper arm initially. As the disease progresses, it affects the skeletal muscles and the other areas of the body. This disease does not impinge on life expectancy, and 85% of affected individuals do not become wheelchair-bound. Next, you have your myopathies, also characterized by muscle weakness and wasting that many may or may not be inherited. These are commonly associated with metabolic diseases and aberrant mitochondrial function. Biopsies of the muscle groups do not display the presence of fibrous and fatty connective tissues, although lipid droplets may be present within the affected skeletal muscle. Desmin-related myopathy. It is a rare inherited disease in which mutation is responsible for the formation of desmin molecules that form desmin aggregates rather than the normal desmin filament. The absence of filamentous desmin results in disorganized myofilaments and skeletal muscle fibers. Disease begins as progressive weakness in the muscles of the legs, followed by weakness in the trunk and the rest of the body. Next is your ethanol myopathy. It occurs as a result of binge drinking, characterized by destruction of skeletal muscle fibers or rhabdomyolysis associated with myoglobinuria or myoglobin in the urine. Muscle biopsy present with swollen degenerating cells and phagocytosis of damaged muscle fibers. Next is your drug-induced myopathy. It occurs as a result of steroid use, either for medical or athletic reasons. Usually, only white and intermediate muscle fibers atrophy, but in severe cases, red fibers may also be involved. Example is chloroquine, which is formerly used for malaria treatment. Rigor mortis, post-mortem rigidity appearing as hardening of skeletal muscle caused by inability of muscle cells to ATP. Under ambient temperature, it begins 4 hours after death, reaching its greatest rigidity about 12 to 14 hours after the person died. Within the 60 to 75 hours after death, the myofilaments begin to decompose and the rigidity of the muscle begins to weaken. Change in the rectal temperature of the body, or algor mortis, in comparison to the normal body temperature is utilized by forensic pathologists to determine the number of hours that the person has been dead. Next is your amyotrophic lateral sclerosis or ALS or Lou Gehrig disease. It is marked by degeneration of motor neurons of the spinal cord and upper motor neurons of the brain. It is an autosomal dominant inheritance. Small persons of affected individual. In some cases, 2 superoxide dismutase gene on chromosome 21. It may involve GTPase regulatory system. It has glutamate toxicity and spinal cord display decreased number of motor neurons. The brain studies shows decreased number of motor neurons in motor nuclei of trigeminal 5 nucleus ambiguous and hypoglossal or CN12 nerves. Other symptoms are dropping objects, loss of manual dexterity, jerky hand and arm movements, advanced spastic motor movements, recurrent pulmonary infection. Late symptoms are your failure of muscle respiration. Next is your myasthenia gravis. It is an autonomic disease which antibodies block the acetylcholine receptors of myoneural junction. This results to reducing number of the sites for initiation of sarcolemma depolarization. It affects men over 60 and women over 40 years of age. It is characterized by gradual weakening of skeletal muscle, especially in most active areas like the muscle of the eye, tongue, face, and extremities. Clinical signs: you have your thymic hyperplasia or thioma, and the presence of circulating antibodies to acetylcholine receptor. Last is the botulism or the food poisoning, which is a form of food poisoning. It is ingestion of Clostridium botulinum toxin, which inhibits acetylcholine release at myoneural junctions. It is marked by muscle paralysis, vomiting, nausea, and visual disorder, and fatal if untreated. Next is cardiac muscle, which is striated involuntary, present exclusively in the heart, originates in the splenopleurotic mesoderm, supplied by your autonomic nervous system, such as your sympathetic and parasympathetic. For microscopic structure, it consists of short and thick branching of muscle fiber. It is intercalated disc, which is a specialized junction that has gap junction and desmosomes. It acts as functional syncytium. The nucleus is centrally placed and it is oval. It has faint transverse lines and is supplied again by sympathetic with the autonomic nervous system. General features of cardiac muscle: contract spontaneously and display rhythmic beat, which is modified by hormonal or neural stimuli. It has one centrally located nucleus, occasionally two, contain glycogen granules, either pole of nucleus. Sarcoplasm is rich in myoglobin, and cells may branch at ends to form connections with adjacent cells. It has thick and thin filaments, poorly defined myofibrils, and exhibit a cross-binding pattern identical to skeletal muscle. It has limited ability to regenerate. Most injuries are repaired by formation of fibrosis or fibrosis connective tissue or scar tissue by fibroblast. Structural component of cardiac muscle cells. They differ from those skeletal muscle follows. T-tubules are larger than in skeletal muscle. That it is lined by external lamina. It invaginates from sarcolemma at Z-disc, not at AI junction as in skeletal muscle. SR is poorly defined and contributes to formation of dyads. Corbular sarcoplasmic reticulum is rich in calcium release channels at the junction feet, and that is analogous to your S-L-terminal cisternae of the skeletal muscle. Movement of ions. First, calcium entry during relaxation. Some calcium slowly leaks into the sarcoplasm through slow sodium channels, contributing to heart's autonomic rhythmic contraction. Calcium also enters cardiac muscle cells from extracellular spaces via voltage-gated calcium channels in T-tubules. The trigger for contraction is the influx of calcium into sarcoplasm that stimulates further calcium release from SR through ryanodine receptors, results in muscle contraction. The strength of muscle contraction. The force is the force of contraction that depends on the amount of calcium available in the sarcoplasm. Normally, about 50% of troponin C sites are occupied by calcium. Now, for calcium removal relaxation. After contraction, calcium is pumped back into SR, mainly regulated by phospholamban. Inactive phospholamban inhibits SR calcium pump. Then, your phosphorylated phospholamban via CAMP-dependent protokinase activates the calcium pump, allowing relaxation of the heart muscle. For mitochondria, more abundant in cardiac muscle and lie parallel to eye bands and are often adjacent to lipids. For the atrial granules, it is present in the atrial cardiac muscle cells and contain precursor of atrial natriuretic peptide or ANP and beta-type natriuretic peptide or BMP, both of which acts to decrease absorption of sodium and water in the kidneys that decreases blood volume, thus decreasing blood pressure. Intercalated disc complex is a complex step-like junction forming end-to-end attachment between adjacent cardiac muscle cells. A transverse portion of intercalated disc. Your fasciae adherentes is analogous to zonal adherens. Your desmosomes are like the muscular adherentes, and it has also the gap junction. For thin filament, these are secured to Z-disc by alpha-actin and binebulet. Your connective tissue elements supports rich capillary bed. The Peyronie fibers are modified in cardiac muscle cells and and bundle of His. It is for conduction and contains few peripheral myofibrils. These are large pale cells rich in glycogen and mitochondria. Clinical consideration: myocardial infarction or MI. This is an irreversible necrosis of cardiac muscle cells due to prolonged ischemia lasting approximately 20 to 40 minutes. Lack of oxygen results to oxygen deprivation. That again results to cardiac muscle which aerobic, thus anaerobic glycolysis. Your anaerobic glycolysis does not produce enough ATP or creatine phosphate and causes accumulation of lactic acid and other noxious substances. If treated within 30 minutes and this ischemia is reversed, cardiac myocytes can recover. 2 to 4 hours incomplete severe ischemia affects cells and it dies, and region of heart will no longer function. Indicative of myocardial infarction, especially last. First is creatine phosphokinase, second creatine phosphokinase, MB isoenzyme, and cardiocyte specific TNI. Cardiomyopathies are diseases of cardiac muscle caused by abnormalities of cells rather than from ischemia. Peripartum cardiomyopathy occurs late in pregnancy and early postpartum period. It is believed to have multiple causes rather than single cause. Signs are elevated BP during pregnancy, nutritional deficiency, immune reaction, and other metabolic disturbances. Now for smooth muscle, it is a spindle-shaped elongated. It is about 30 micrometer in length but can grow as 200 to 500 microgram in pregnant uterus. It is non-striated and involuntary. It is supplied by ANS, and adjacent smooth muscle cells are in contact with each other through gap junctions that help transmit electrical impulses from one cell to another. The nucleus are centrally placed. Structure of smooth muscle cells. Nucleus, again, centrally located. It is in longitudinal sections and can have corkscrew shape or deeply indented. For cytoplasmic organelles, the mitochondria, RER, and Golgi apparatus are concentrated near nucleus and involved in synthesis of type 3 collagen, elastin, glycosaminoglycans, external lamina, and growth factors. Your sarcolemmal vesicles or cavillae are seen in periphery of your smooth muscle cells. It may function in uptake and release of calcium. Your ECRs are sparse and aligned obliquely to longitudinal axis of your smooth muscle cells. Filaments of smooth muscle. Contractile filaments, you have your actin and myosin. It is not organized into myofibril and they are attached to peripheral on cytoplasmic densities and are aligned obliquely to longitudinal axis of smooth muscle cells. For thick filaments or your myosin 2, each surrounds as many as 15 filaments. That prior to contraction, myosin 2 molecule is inactive and can't bind to actin filament because the tail of myosin molecule is attached to heavy meromyosin. Thin filaments like actin, caldesmon, tropomyosin, and calponin. Your caldesmon with tropomyosin masks sites where myosin binds to affect muscle contraction. For your intermediate filaments, they are attached to cytoplasmic densities and include vimentin and desmin in vascular smooth muscle, desmin only in non-vascular. Cytoplasmic densities, which is analogous to ZDIS, contain alpha-actin, actinin, and various ZDIS associated proteins, and functions as filament attachment sites. The gap junctions are between the smooth muscle cells and facilitate the spread of excitation. It is collectively called a nexus. Contraction of smooth muscle cell. More slowly and lasts longer than skeletal muscle because the rate of ATP hydrolysis is lower. Contraction is stimulated by transient increase in cytosolic calcium. First, calcium binds to calmodulin and alters the conformation. Your calcium-calmodulin complex activates the myosin light chain kinase that catalyzes phosphorylation of regulatory light chain of myosin. Now, your myosin activation and actin interaction. The phosphorylated myosin unfolds and forms thick filament, producing calcium and then resulting to calcium removal, and calcium removes inhibitory effects of caldesmon and calponin, allowing actin-myosin interaction. Now, contraction and ATP use. Myosin heads bind with actin, activating myosin ATPase, leading to contraction. Cycle continues as long as myosin remains phosphorylated. For relaxation, Myosin dephosphorylation disrupts actin binding, causing relaxation. Initiation of contraction for your vascular smooth muscle is triggered by nerve impulse with little spread of impulse from cell to cell. For visceral smooth muscle, stretching of muscle itself is myogenic, signals spread from cell to cell. For uterus, it is triggered by oxytocin and epinephrine for elsewhere in the body. Innervation of smooth muscle. It is by sympathetic or non-adrenergic nerves and parasympathetic for cholinergic nerves of your autonomic nervous system, which act as an antagonistic function or to stimulate or depress activity of muscle. Now let's go to clinical consideration. Leiomyoma is a benign neoplasm of smooth muscle. It is frequently present in fetus, where it forms most common benign tumor in women. Additional locations is your nipple, breast, nipples of breast, erector pili of muscle skin or the pilar leiomyomas, labia majora, scrotum or eugenectal leiomyomas. These are small individual tumor, less than an inch size, and occasionally displays mitotic figures. Surgical excision is normal unless tumors are numerous. Leiomyosarcoma is a compact, painless, malignant tumors of the smooth muscle. It is more common in adult female, and almost 20% of sarcomas are of non-calcified tissue. Skin, deep tissues of arms and legs, retroperitoneal, soft tissue, and abdominal cavity. Now, differences between skeletal, cardiac, and smooth muscle. Skeletal, for the muscle fiber, has very long and branched cylindrical. For cardiac, they are small, branched and cylindrical. For smooth muscle, they are small but branched, spindle shaped. For nuclei, for skeletal muscle, it's multinucleated, peripherally placed, or flat. For cardiac muscle, it is centrally placed, single, and oval. For smooth muscle, it is single, centrally placed, and elongated. For striations, skeletal muscle has well-defined cross striation. For cardiac, it is poorly defined, and for smooth muscle, it has no striation. For your SR, skeletal muscle has triads, again, two cisternae, terminal cisternae, and one T-tubules in the middle. For cardiac, it forms dyads, one T-tubules and one tubular cisternae. For smooth muscle, it is not well organized. For T-tubules, skeletal muscle, it is present in the AI junction, while it is present in Z-line in the cardiac muscle. It is absent in the smooth muscle. For cell junction, it is absent for skeletal muscle. For cardiac, you has your intercalated discs. For smooth muscle, you have gap junctions. For skeletal muscle, you have voluntary, while they are involuntary in cardiac and smooth muscle. For the innervations, both your cardiac and smooth muscle are innervated by the autonomic nervous, while the skeletal is innervated by the motor nerves. For mitosis, for skeletal and cardiac, they have none, and it is present in smooth muscle. Regeneration after injury, for skeletal muscle, it is limited, but is not available in cardiac muscle, but also present in smooth muscle. Locations, skeletal muscles in tongue, esophagus, and diaphragm. Your cardiac, you have your heart, superior and inferior vena cava, and pulmonary veins. For your smooth muscle, you have your vessels, tubular viscera, and organs. Now, contractile non-muscle cells. First is your myoepithelial cells. Location and attachment is found in certain glands, sharing basal lamina with secretory and duct cells. It is attached to basal lamina via hemidesmosomes. For structure and function, it is a basket shape with radiating processes resembling smooth muscle cells derived from ectoderm, where they contract to put secretion into ducts. Composition, it contains actin, myosin, and intermediate filaments connected to cytoplasmic and peripheral densities. It contracts via calmodulin-mediated process similar to smooth muscle. For hormonal response, oxytocin stimulates contraction in lactating mammary glands, and acetylcholine triggers contraction in the lacrimal glands. For myofibroblast origin and composition, it is derived from mesenchymal cells and contains vimentin, caldesmon, and cytokeratins. Its functions are it resembles fibroblasts but have more actin and myosin, enabling contraction. It plays a key role in wound healing and by contracting to reduce wound size. For tonicity, it is the passive muscle contraction known as residual muscle tension, not the same as achieving muscle tone through exercise.